Hello, everyone, and welcome to today's broadcast, Discovering Cellular Mechanisms and Markers in Anti-PD1 Non-Responders through Ingenuity Pathway Analysis and Oncoland, presented by Dev Mistry, Senior Field Application Scientist, Bioinformatics, QIAGEN. I am Alexis Krause of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by QIAGEN. For more information about our sponsor, please visit QIAGEN.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Mistry. I will now turn the presentation over to him.
Thank you for the kind introduction. So today we'll be looking at two different software. One is Ingenuity Pathway Analysis and another one is Oncoland from Omicsoft. And we will learn how this software can be used to discover cellular mechanisms and biomarkers in pretty much any data set, but for this presentation we'll focus on anti-PD1 non-responder data set from renal cell carcinoma. In terms of agenda, the main objective for the study is to identify biomarkers, regulators, targets, and mechanism of action from an NGS dataset. Now, this dataset can, again, be a user dataset or publicly available dataset. In this case, we're going to use a publicly available dataset for anti-PD1 non-responders. This presentation is divided into two parts. In the first part, we'll be focusing on data mining. So we'll be using Oncoland from Omicsoft for data mining. So I'll introduce you to the software. I will find a good data for anti-PD1 non-responder from this software. And then we'll study how the software can be used to identify biomarkers by looking at the expression across different tumor tissue from public sources such as TCGA, GTAC, and so on. And then in addition to that, this software can also be used to generate different views. So to see how good the biomarkers are, you can generate curves such as survival curves, heat maps, and so on. In the second part of the software, we'll be focusing on data interpretation. And in this part, we'll be looking at Ingenuity Pathway Analysis software. So I'll introduce you to the second software and the database backing the software. And then Ingenuity Pathway Analysis is mainly used for interpretation. So we'll look at, like, okay, if you have a gene expression data, how this gene expression data is associated with pathways, master regulators, and how it can also be used to discover biomarkers and doing comparisons. And then we will end with a summary. So say you're using your own data set. In this case, what you'll be doing is you'll be using pipeline that is offered by RStudio by QIAGEN. And this pipeline will allow you to do things like pre-processing the data, QC the data, aligning your read to the reference if you're using like RNA-seq or NGS data, and then gene transcript quantification. And then finally, if your data has experimental groups like disease versus normal, treated versus untreated, then you can use statistical tools available within the software to do comparison. And once you do all that, you can generate various plots that are shown on the screen over here. So you can generate principal component analysis and principal coordinate analysis plot. You can also generate volcano plot, which is in the center of the screen. And you can also generate heat map, which is on the lower right corner of the screen. Now, many users, not only they use their own data set, but they're also interested in locating interesting public data sets. And if you're one of the users like that, then what you would probably have to do is you would have to download the data from public domain. So this data can be gigabyte, if not terabytes. After you download, you would have to align the reads to the reference for this data if this is an RNA-seq NGS data. And then you would quantify gene and transcript expression, you would do differential expression as I mentioned in my previous slide. So you would have to do all of this by yourself. Now, if you're interested in curating multiple public data from multiple sources, then in addition to doing all of this, you would also have to curate metadata for this data. So each data set will have description, right? Where is it coming from? What tissue is it coming from? What cell line is it coming from? If it is coming from a patient, then what was the patient treated with? What was the concentration of drug? So you would have to curate all of this information. Fortunately, our team at Omicsoft has already done this from many different public sources. So this will be sources such as the Cancer Genome Atlas, TCGA, which has a lot of cancer data set. GEO, CCLE has a lot of cell line data set. GTEx, which has a lot of normal tissue data set. So our team has already done all of this process for you, which can take months, if not years, depending on the resources you have. And another thing our team is regularly doing is they're regularly maintaining this database, and they're also updating this regularly. They're updating this quarterly. So now, say you're a user, and you are focusing on something like PD1, PD-L1 interaction. For those of you who don't know PD1, PD-L1, this interaction is one of the immune checkpoints. PD-L1 is expressed on certain tumor tissue, and it is known that this interaction can suppress the immune system. So that's why there are so many therapies that are focused on blocking this interaction. So one of the antibody drugs that is out there is nivolumab from BMS, and it is known to block PD1, and it is also known to be used in indications such as melanoma, non-small cell lung carcinoma, renal cell carcinoma, and other indications. Now, the problem with such treatment is it is beneficial for many patients, but it does not apply to all the patients. So there are many PD-L1 expressing tumors that don't respond to the therapy. So that's why I decided to pick a data set which compares non-responder population to responder population. So now the question is, I introduce you to Omicsoft LAN database. How can we use this LAN database to locate such a data? So as I mentioned, this LAN database has curation from many different public sources. And one of the sources, if you look at the left-hand corner of the screen, is GEO. GEO has tons and tons of public data set, and these are curated in the LAN database. So since we have curated all of this data, you can actually search for them by different keywords. You can use keywords such as disease names, drug names, etc. So over here, I have a screenshot from the software, which you can see. And in the screenshot, you can type in the name of the drug. So in the previous screen, we have nivolumab. So you can start typing nivolumab, and some hits will come up. If you click on any one of the hits, then you can actually locate different projects that have used nivolumab in their study. So at the bottom of the screen, I have a list of studies that have used nivolumab. And since we have also curated the metadata from the study, you can see what institutions are the study coming from. You can also read the general description of the study. And more importantly, in the bottom center of the screen, we have some web links to the study. So you can click on the web links and you can read actual abstract, data description, and so on. So that's what I did. I actually read through the description of the studies, abstracts of the studies, and even some papers. And then I realized that the project that's listed over here, the ID GSE 67501, it's kind of highlighted in the center of the screen. It was an interesting project. Interesting why? Because in this study, investigators are working with patients that have metastatic renal cell carcinoma, and they are treated with nivolumab. Now, these patients are classified as responders and non-responders based on response evaluation criteria in solid tumors. And then what authors have done from this patient is they have extracted RNA from PD-L1 plus tumor tissue. So they have extracted that from 4 responders and 8 non-responders. And then later on, they have performed a microarray analysis on it. So they have performed gene expression analysis using Illumina bead chip. Some of you may be interested in checking out this paper, so I have included a screenshot at the bottom. So please feel free to check it out. Now, one of the things we are focusing on is biomarkers and targets. So if you want to look at that, then you might be curious, how you can open up this gene expression data and look at different biomarkers and targets. So over here, we have opened up this data set in form of volcano plot. And volcano plot is in the center of the screen. And on the right-hand side, you also have this data available as a differential expression table. And in differential expression table, what you see is a list of genes with fold changes, p-value, adjusted p-value, and so on. And in volcano plot, what you see is on the x-axis, we have log fold change for different genes. So if you go from 0 to right-hand side, you have genes that are increasing in expression. And if you go from 0 to left-hand side, you have genes that are decreasing in expression. Also, if you look at the y-axis, we have anti-log of p-value. So if you go from 0 to upward on y-axis, then you have genes that are more and more significant. So if you think about it, genes that are present in the upper right corner of the screen, these are going to be the genes that are upregulated and significant. And then genes that are present in the upper left corner of the screen are going to be the genes that are downregulated and significant. So now let's see if we can focus on non-responder population. So again, if we look at the volcano plot, this volcano plot is comparison of responder versus non-responders. So if you focus on the upper left corner of the screen, then you'll see genes that are genes that are upregulated in responder and significant. And this paper particularly mentions UGT1A6 as one of the key genes because this gene is involved in metabolic and solute transport functions. So if you kind of read about this gene online, then you'll know that this gene, what it does is it processes steroids, drugs, etc., into water-soluble molecules that can be excreted from the cell. And this is what the paper is proposing, that this is one of the main mechanisms by which non-responder may not be responding to therapy. But in addition to UGT1A6, there are other genes of that family that are present in non-responder population as well. So far, what we have seen is we have introduced you to Oncoland database. Oncoland database has information from many different public sources such as TCGA, SRA, GEO, and so on. We have also introduced you how this database can be used to locate interesting data. So in our case, we have located data which is responder versus non-responder. And then now what we're going to look at is, can we study different biomarkers using different information that's available in this database. So for this, now we're going to focus on UGT1A family members because that's one of the things that's described in the paper. And in the later part of the presentation, we're going to move away from UGT1 and we're going to focus on other genes as well that are not described in the paper. So last time we looked at Oncology database. This time we're going to look at the Cancer Genome Atlas database. And in this database, the data we have curated, what you can do is you can search for the gene or marker of your interest and see how this gene is expressed across different tissues. So on the y-axis, we again have gene expression. And on the x-axis, we again have gene expression. And on y-axis, we have different tumors. So what you see over here is UGT1A6 is expressed more in carcinomas such as renal cell carcinoma and hepatocellular carcinoma, and it's expressed relatively less in carcinoma such as acute myeloid leukemia. So in some ways, it does seem to be expressed more in renal cell carcinoma compared to other tumors if you think about its marker application. Now, the thing is, not everyone is going to be interested in all of these diseases. Many times people want to focus on specific diseases. So for example, this study is based on renal cell carcinoma. So within the software, we also have filters which you can use to focus on specific indications. In this case, I have only selected one indication, renal cell carcinoma. But in your case, say if you wanted to select multiple indications, like if you wanted to compare renal versus acute, then you could select more, and you can only have those rows visible in the software. Another thing you'll notice is on this plot, we have two types of colors, blue and green. So blue are the different tumor samples that are curated from TCGA, and green are different normal samples that are curated from TCGA. So another thing a lot of users are interested in doing is splitting this row based on the metadata. So in this case, the metadata would be tumor versus normal. It is also very easily doable in the software. you can change the grouping by a few clicks, and you can split this one screen into split this one row into two rows. So now we see tumor versus normal comparison. And you can see difference is not that great, but there does seem to be some difference between tumor and normal tissue. So in this case, another thing we can check on is over here, tumor is just like one thing, but in reality, there are different stages of tumors. And this metadata is also available in TCGA. And because this is available in TCGA, because we have curated it, you can now split that single row of tumor into multiple row of stages. So now we have pathological stage 1, 2, 3, and 4. And again, the pattern you'll notice is there's some difference between tumor and normal. And the difference between stages, honestly, is minimal. But this is only for one marker, UGT1A6. What about other family members? So now when we move on to another family member, UGT1A3, you can see there's bigger difference between its expression in tumor and its expression in adjacent normal tissue. So keep in mind that this is TCGA, so normal over here is the adjacent normal tissue. And then again, just like UGT1A6, if we split this into different stages just to see if this marker is specific to one stage or more, what you'll see is there is bigger difference between normal and stage 1, but there's smaller difference across different stages. But at the same time, if you go from normal to stage 4, then the difference is becoming greater and greater, giving it potential as a biomarker. So as I pointed out in one of the previous slides, the normal tissue we've been looking at so far is normal that's adjacent to tumor. And a lot of investigators don't like that about TCGA because they feel that the normal tissue that's next to tumor may or may not be normal. So what they want to do is they want to compare the expression of this gene from tumor samples from TCGA and normal samples from GTEx database, which is solely for normal tissues, okay? And because our team has diligently curated information from all of these databases, you can actually do this cross-database comparison. And another thing to note is you are only able to do this comparison because our team has gone through trouble of reprocessing all of this expression data using the same pipeline. So now I have similar plot, but now we are looking at two different databases. We have TCGA on the top and we have GTEx at the bottom. And what you see is there is difference between expression of UGT1A3 between tumor and normal in TCGA. And there is lesser difference between normal of TCGA and GTEx. So overall, there's difference between expression of UGT1A3 of normal GTEx and TCGA and tumor of TCGA. And on the lower right corner of the screen, you can see I have also turned on the statistical test, which is ANOVA F test. And according to this, the difference we see across groups is significant. So this is another thing a lot of users like to do when they're using Oncoland. They like to turn on the statistics and see if the difference they're looking at is real or not. So summary for so far is we looked at two markers, UGT1A3 and UGT1A6. We looked at these two markers because according to the non-responder versus responder study, these markers are more prevalent in non-responder. And another thing we noticed is for UGT1A6, there was less difference. For UGT1A3, there was more difference when we compared between tumor and normal. Nevertheless, when you compare across different tumors, UGT1A markers are expressed more in renal cell carcinoma compared to other tumors. And another thing I did not go over much, but you as a user can do, is you can go beyond TCGA and GTEx. You can also use databases such as CCLE, which I have highlighted in the center of my screen. And using CCLE database, you'll see similar information in the sense that UGT1A6, which is one of the markers, is expressed more in kidney cell line compared to other cell lines such as, say, lung or breast and so on. So now, we can ask another question. Are these markers associated with survival? And because the study is emphasizing greatly on UGT1A6, I have picked that marker. So another thing you can do in Oncoland software is you can divide things into groups. So the way I have divided information over here is I have divided samples into high expression of UGT1A6 marker, which is the top row, and low expression of UGT1A6 marker. So I basically define a cutoff of 50%, and I have mentioned to software that, okay, all the samples that have 50% or higher expression, make them high, and all the samples that have 50% or lower expression, make them low. And even though you might think that there are more samples in high versus low, it's not the case. In low, things are just clustered very close to each other. So there are about equal number of samples in high group and low group. So within software, once you group this, very easily by one or two clicks, you can also generate survival curves. And this is something I know is of great interest to many of our users. So what they want to do is they want to compare the survival when UGT1A6 or marker of the interest is high versus marker of the interest is low. And in this case, what you see is when you have high expression of UGT1A6, then you have relatively poor survival, as opposed to that when you have low expression of UGT1A6. Now, this is across all the tumors in TCGA, as opposed to just renal cell carcinoma. But at the same time, you can see, if you look at the statistics, we have a significant value if you focus on the upper right corner of the screen. Now, if we limit this to just renal cell carcinoma, so again, regroup samples based on high and low expression only in renal cell carcinoma, and then generate survival curve within the software, then you will see similar pattern, which is samples that have high expression of UGT1A6 will have poor survival, compared to that samples that have low expression of UGT1A6 have better survival. So, I mean, one can always ask the question that is UGT1A6 is actually associated with survival or not. So in the last two slides, to summarize what we learned is within the software, you can generate groups. So in this case, we generated groups based on expression of marker, UGT1A6. And what we learned is overall, when you have higher expression of this marker, then there is poor survival, whether you're looking across all tumors or whether you're looking across renal cell carcinoma. Now, what I want to mention over here is this is just one way of dividing data based on expression marker, but you can also divide data based on other parameters. So we have curated metadata, right? So we also have curated histological data. So based on that histological data, you can also divide tumors into, say, HER2 positive versus HER2 negative, or you can do triple negative breast cancer versus non-triple negative breast cancer. Some people, instead of working with expression markers, they work with mutational markers. So they would divide data into TP53 wild type versus TP53 mutant. The key is once you divide samples into this group, you can generate wide ranges of plots. So in the lower left corner of the screen, I have survival plot. But in addition to that, once you divide things into groups, you can also generate gene expression heat map. So in the lower right corner of the screen, I have an example of gene expression heat map, which compares TCGA tumor tissue to both TCGA and GTEx normal tissue. So TCGA and GTEx normal tissue is clustered around the center of the heat map, where you see a lot of red, which means the genes are going up, and the same genes are going down in tumor tissue. So you can also come up with good markers by generating such heat maps. So in part one, we focused on data mining. More specifically, we learned about the LAND database. We learned how it can be used to locate a data set of interest. In our case, it was the anti-PD1 non-responder data set. And we also learned how it can be used to study various biomarkers and targets. In this case, we have mainly focused on UGT1A6 and UGT1A3 because these are two of the enzymes that are mentioned in the paper, but we can go well beyond that as well. Now, in part two, we are going to again focus on data interpretation. So for data interpretation, I'm going to first introduce you to Ingenuity pathway analysis software and its database. And then we are going to go into things like how it can be used to learn about mechanism of action in non-responder, how it can be used to generate causal networks in non-responder, how it can be used to discover potential new biomarkers and targets, both through looking at pathways and through comparison analysis. So when we looked at that volcano plot earlier, right, that volcano plot had many genes. They did not just have UGT1A6, but they had many genes. that are going up and down. And one can always ask the question, okay, you just spent half the presentation talking about one gene. What about the other genes? So this is where we can actually use software such as Ingenuity pathway analysis effectively. What Ingenuity pathway analysis will do is it will actually take the whole gene list, and based on that, it will show you what pathways are enriched for that gene list. So what pathways are associated with that gene list, what diseases and biological processes are associated with the gene list. If you look at the right-hand corner of the screen, you can actually open up the pathway. If you look at the chart where you see left one, you can also compare expression of different biomarkers on the pathway. And if you look at the bottom of the screen, then you can compare your various data sets, and you can also compare your data set with public data set in the sense that you want to know what pathways are active and inhibited across all of this data set, what diseases and functions are active and inhibited across all of this data set. So these are the things we are going to do. But all of these things are only possible because of the database that's backing IPA, and this is what we call knowledge base. So in this knowledge base, our PhD level curators, what they have done is they have curated information from peer review publications, and they have also curated information from third-party data sources. Now keep in mind, a lot of this information that is curated from peer review publications is manually curated by PhD level scientists, and it is updated on weekly basis. On the other hand, the data, the information we curate from databases like BioGrid, clinicaltrials.gov, Tarbase. So, these are the databases for things like protein-protein interaction, clinical trials, microRNA. This information is updated on quarterly basis. So all in all, our curators keep this database as up-to-date as possible, so you get as up-to-date analysis as possible. Now, keep in mind, we mainly curate information from human, mouse, and rat, but saying that if you're working with other species like bovine, chicken, chimpanzee, you can still use IPA. As a matter of fact, if you go to scholar.google.com and search for IPA with this other species, you'll find plenty of publications. One thing that is also unique about this database is you're giving it a gene or gene list, and what it's doing is connecting these different genes and proteins. So in addition to making those connections, it will actually give you a lot of contextual information. So over here, I have on the right-hand side this chart connecting ILK protein to PDK1 protein. And what you see over here is there is an edge connecting them, which says PP bracket 3. So PP stands for protein-protein interaction. So first of all, just by looking at a network or pathway, you'll know that this is a specific type of interaction. And 3 stands for number of sources that support this interaction. In addition to that, by a few clicks, you can go from that line to the findings that's in the upper right corner of the screen over here. And looking at the findings, you can quickly see what literature is this interaction coming from. You can click on the hyperlink and go to the literature itself. You can also see what type of experiments did the author do to derive this information. You can see what species they use, human, what cell lines they use, human plateauate. So the information we have in this database is very contextual. And just to draw contrast from the previous database, which is the Oncoline database, in Oncoline database, our curators have curated actual NGS data. As opposed to that, in IPA database, we have curated findings or knowledge. So basically in IPA database, our curators are reading through papers, and then they are collecting information from text and figures. As opposed to that, in Oncoline database, we have curated actual data. So that's one of the biggest differences between these two databases. And that's why these are very complementary to each other. So in the last few minutes, what I have done is I have introduced you to Ingenuity Pathway Analysis database, and I have also drawn contrast with Omics database. And again, the contrast is in Omics database, we have curated actual data. In IPA database, we curate knowledge from text and figures. So before I start showing you pathways in IPA, I want to go through some color codes. So over here, you see a couple of different colors, and I want to emphasize on 4 of them. 2 are red and green, and other 2 are orange and blue. Red and green are actual measurements. So red are the genes and proteins that are going to be upregulated in your data set. Green are the genes and proteins that are going to be downregulated in your data set. As opposed to that, orange and blue are predictions. So orange are the things that are predicted to be active according to IPA, and IPA is drawing this prediction based on the information it has collected from previous literature. And then blue are the things that are predicted to be inhibited. So now if we walk through some examples, we see MST1 in the center of the screen connected to apoptosis. MST1 is red. That means it's upregulated in the specific gene expression data set we are studying. And this is leading to increased apoptosis. I'm saying increase because apoptosis is colored orange. So MST expression is the actual data. Apoptosis, orange, is the prediction. As opposed to that, if you look at the center of the screen, bad is colored green. So bad is downregulated in your data set. And this is leading to inhibition of apoptosis because apoptosis is colored blue. And then finally, just to give you one more example, just to make sure we get these colors down, TGF-alpha is colored red. That means TGF-alpha is upregulated in the specific data set we are looking at. And ERBB2 is colored orange. That means upregulation of TGF-alpha leads to activation of ERBB2 according to information that IPA has curated from previous literature. Okay? So again, red and green, actual measurements, orange and blue, prediction. Now that we have this down, let's look at pathways. Excuse me. So let's look at pathways. So over here, what we have is pathways that are enriched in non-responder population. So what I have done is I have taken that Omicsoft data and I have uploaded non-responder versus responder comparison in IPA. Okay? And then if we look at the name of the pathway, you should immediately see relevance of some pathways. For example, second to top pathway is molecular mechanism of cancer. In non-responders, we expect cancer to be on, and that's why that pathway makes sense. In addition to that, there are also pathways such as DNA methylation and transcription repression signaling in the center of the screen. And in cancer, such things are supposed to happen, so we can expect that as well. Now, some other pathways may or may not make sense. For example, there is axonal guidance signaling on the top, and you would think that, hey, this is a renal data set. Why do we see that pathway? But the pathways that don't make sense, they're like kind of colored in blue over here. These pathways are related to functions such as cell growth, growth factors, inflammation, cell cycle, and all of these things are related to cancer. So it makes sense that these pathways show up as top hit for non-responder data set. What we can do to study mechanism of action in non-responder is we can actually open up one of the pathways. So in this case, in software, I'm clicking on molecular mechanism of cancer, and I'm opening up that pathway. And when we open up the pathway, if you're looking at IPS pathway for the first time, one thing you'll notice is pathways are very nicely organized from plasma membrane to cytoplasm to nucleus. And for this specific pathway, we have plasma membrane on the both sides of the screen, on the top of the screen as well as bottom of the screen, right? So let's zoom into this pathway and learn more about it. What's happening? So again, remember the color. Orange and blue are prediction, red and green are actual measurement. If you focus in the very center of the screen where it says DNA repair and go 2 steps down, then we have a gene that is colored in red. So red means it is upregulated in the data, in your data set. So this gene seems to activate gene that is downstream of it, and that activation seems to lead to increased DNA repair. In addition to that, we see that differential expression of other genes seems to lead to increased cell cycle regulation and decreased apoptosis if you focus on red circles that I have drawn on this pathway. So now we have bigger picture. Now we are not like looking at just one gene, we are looking at multiple genes, and we are learning how these multiple genes are leading to things like increased DNA repair, increased cell cycle regulation, and reduced apoptosis. And all of these things will be, of course, important in non-responder population. And then if we look at the bottom half of the pathway, then again, we have left one color red, and that seems to lead to increased cell survival. And this is the section that I have highlighted in red in the center of the screen. And I'm going to talk a little bit more about left one in the following slides. So what we learned so far is using these pathways that are provided by Ingenuity pathway analysis, we can see how gene expression in your data set is connected to specific processes such as DNA repair, apoptosis, cell cycle regulation, and so on. Now, we have two groups over here, right? We have non-responders and we have responders. So can we compare those groups in IPA? So that's the question we are asking over here. So in this case, you can see what you can do in IPA by a few clicks is you can turn on this bar chart. So next to left one, I have this bar chart, and I have group number 1 and group number 2. Group number 1, by the way, is non-responders, and group number 2 is responders. And you can see left one is up in non-responder, and left one is down in responders. And if you know about left one is a very well-known marker for cell survival, and it has been known to be important for cell survival for many different cell lines. And as expected, it is up in non-responders, and it is down in responders. But this is a well-known marker. What about other markers? So in IPA, for specific pathways and networks, you can also generate heat maps like the one I have shown on the right-hand side of the screen. So now we are not just looking at left one, but we are looking at many other genes such as CC, ND2, PSEN2, and many others. So all of these genes are up in non-responders and down in responders. And I'm sorry I have switched the column header, but red column is the non-responder and green column is the responder. So now we can also potentially propose other biomarkers that may be specific to responders and non-responders. And then next thing we can ask is, okay, we were doing comparison on a single pathway. Can we do more for global comparison? Can we compare multiple pathways at the same time? So in IPA, you can actually do that. So over here, I have compared activation and inhibition of all of the pathways in non-responder versus responder. So the first column in the center of the screen that's orange is non-responder, and the second column is responder. And you can see a lot of cancer-related pathways are active in non-responder, and they are inhibited in responder. So this can also help you propose a hypothesis why non-responders are resisting the therapy. And even though I have opened up a different pathway, in this case, it's IL-7 signaling pathway as opposed to molecular mechanism of cancer pathway that we saw in previous slides, you see similar conclusions that apoptosis is off, bottom of the screen, proliferation is off, bottom right of the screen, and on the left-hand side of the pathway, you also see altered metabolism, which is also one of the characteristics of the cancer. So again, it's a different pathway, but conclusions are very similar, like why non-responders are not responding to therapy, because apoptosis is off, cell cycle is on, proliferation is on, and then there is also altered metabolism, which is in agreement with what is discussed in the paper. So one of the other things many users like to do is they like to compare their data set to public data set. So why would you want to do that? So here's the thing. We are looking at only one renal carcinoma study, right? But there are many other studies that have also used renal carcinoma samples. So if you compare your study to this public study, you can maybe strengthen your hypothesis. So for this, in IPA, we have introduced analysis match tool. And analysis match tool can either allow you to compare various analysis that you have. So say you have 30 patients, 50 treatments, you can compare them side by side using analysis match. Or it can also allow you to compare your data with public data, which is what I'm doing over here. I am comparing the data that I found from the study to other public renal carcinoma data sets. Now, what we see is many of the regulators that are found by IPA are similarly activated between the renal carcinoma data set we are looking at, as well as many other public data sets. So this feature is one of the newer features in IPA. It's called Analysis Match, and it's a very powerful tool to compare how different pathways, different regulators, different biological processes are similarly or differentially activated and inhibited across different analyses. So what else can we learn about these regulators and targets? So in IPA, we have this feature called causal network analysis. And causal network analysis feature focuses greatly on master regulators. all right? So there are many columns on the screen right now, but the left-hand side column is the column with all of the predicted regulators by IPA. And on the very right-hand side column where you see a lot of genes, these are the genes that are downstream of these regulators and are present in your data set, okay? So in this case, the data set we are looking at is non-responder data set. So if I were to give you an example of a row, then the very first row where it says CRE-BBP, so CRE-BBP would be a master regulator. The very first row, what is telling me that CRE-BBP is a transcription factor. It is predicted to be activated by IPA, and it is regulating 375 genes in this non-responder data set according to the information that is curated from the literature. So that's what the first row is telling me. And I have actually filtered this table for specific genes. So I have first of all filtered this table for only genes and proteins as opposed to drugs and chemicals. So because IPA can also predict whether genes in your data set can be regulated by certain drugs and chemicals. So I have filtered this table for genes and proteins. And then I have also filtered this table for genes that are significantly up and down by the criteria I have defined, which is the log2 ratio of 0.58, which converts into fold change of 1.5. So when we apply this filter, we actually see many interesting transcription regulators as upstream master regulators. And these are the regulators that are in the center of the screen. And you can see all of these regulators are predicted to be active. And then if I were to open up networks for them, you would see that all of these are also actually connected to activation of renal cell carcinoma. So if you are one of the users who is focusing on mechanism of action, then this can be of great help. And then below all of the transcription factors, we see a couple of enzymes such as GFPT1 and SMYD3, right? So if you're one of the users who are looking for master regulators that are targetable molecules because you are doing things like drug discovery and so on, then you may want to focus on these genes that are enzymes because enzymes are targetable. Not to say transcription regulators are not, but in general, like, my conversation with different pharma and biotech, in terms of small molecules, they prefer something like enzyme as opposed to transcription factors. So all of these activated factors, when I do enrichment analysis on them, they are actually connected with cancer in one form or another. A lot of them are also connected with processes such as cell cycle, DNA repair. And then in terms of this enzyme, one of the ones that piqued my interest was GFPT1. I focus on GFPT1 because in engineering knowledge base, it is curated that this enzyme has been previously associated with diseases such as prostate cancer, obesity, diabetes. And then when we look at the causal network, it provides us with an explanation that how activation of this enzyme is leading to increase in renal cell carcinoma phenotype. So what we see on this causal network is GFPT1, when it's active, it is leading to activation of other regulators such as TGF-beta1, USF2, and PRKAA. And then this activation is leading to activation of downstream oncogenes such as MYC, and then also TP53. And then this is leading to increased renal cell carcinoma phenotype. So now, as a user, you can propose a hypothesis. And if you're interested in discovering target, then GFPT1 can also serve as a potential target. And then finally, before I wrap up my talk, for those of you who are interested in like biomarker discovery, we have tools such as Bioprofiler. And when you use these tools in IPA Bioprofiler, you can actually connect different genes and proteins that are present in the data set with specific phenotypes that you're looking at. So in this case, we are connecting the differential gene expression in non-responder to renal cell carcinoma. And using that, we are figuring out that B2M is one of the markers that's expressed in renal cell carcinoma. It's also transmembrane receptors. And if you're curious, like, where is this information curated from, in IPA, things are very easy. You can either double-click on the lines that are connecting different genes, or you can click on the hyperlink that's on the right-hand side of this table, and you can open up this findings page. So on the lower right corner of the screen, you see finding, and you can figure out, like, what paper or what source is this information coming from. So to summarize, in the second part of this talk, what we did is, first of all, we generated hypotheses for mechanism of action. For that, we looked at pathways such as mechanism, the cancer pathway and IL-7 signaling pathway. And we also looked at causal networks such as GFPT1. All right. So using that, we proposed certain mechanism of action in terms of how renal cell carcinoma phenotype is on in non-responder data set. For biomarkers, we also looked at cell survival markers such as LEF1. And then in addition to that, we also discovered potential new markers that are shown in the heat map on the screen. And then another thing we learned for IPA is it can also be a powerful tool for comparison. So you can compare your data with your other data, but you also can compare analysis of your data with analysis of data sets from public domains such as TCGA, GEO, SRA, and so on. One thing I did not cover is actually shown on the right-hand side of the screen, where I'm using an example when IPA can also be used when you don't have any data set. So in that case, say if you were the author of the paper and you wanted to quickly figure out how UGT1A6 is connected to renal cell carcinoma, then even without any data, you could draw the network that is drawn in the lower right corner of the screen, and you could propose hypotheses. So overall summary for this presentation would be we looked at two software. One is Oncoland from Omicssoft, and second one is Ingenuity Pathway Analysis from Ingenuity. Both Omicssoft and Ingenuity are part of QIAGEN. Both of this software have their own unique database. So in LAND database, our curators have curated information from public sources such as TCGA, SRN, GEO, and so on. And again, they have curated actual data, and they have reprocessed all of this NGS data, especially the gene expression data, using the same pipeline. As opposed to that, in Ingenuity, our PhD level curators, what they're doing is they're curating knowledge. So they're reading text and figure from the journal, and they're curating knowledge. So using this knowledge in both of the software, you can do wide ranges of things. So in Oncoland, we looked at how you can see the expression of a gene across different tumor samples, tumor versus normal samples, different stages of tumor. You could even locate an interesting data set. So that's how we found the non-responder versus responder data set for renal cell carcinoma. And then we also generated plots like survival curves. I did not generate heat map, but that's also something very easily doable in Oncoland. And for IPA, what we learned is mechanism of action. So we opened up different pathways that allowed us to generate hypotheses for mechanism of action in non-responder. We also looked at biomarkers, and we also did some powerful comparison using this tool called Analysis Match. All right. So with that, I will conclude my presentation. And if you guys have any questions, I'm happy to take them right now.
Thank you, Dr. Mistry, for that informative presentation. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. Our speaker will follow up with your questions via email. So let's get started. Our first question is, can you talk about the export options for different views that you showed in your presentation?
Yes, great question. So everything I have shown, right? So this is a concern that's brought up by many of the users when they actually start using our software, that they use some other software in the past and that don't allow export. So pretty much all the views I have shown, they can be exported as high resolution images. And especially for things like Ingenuity Pathway Analysis, if you go to scholar.google.com, you see plenty of publications that cite IPA and use views that are generated by IPA. So all of the views are exportable. They're basically export button on top of the screen. You would just click on it, and then you would choose things like what type of image you want. Do you want JPEG or PNG? And in case of IPA, you can also adjust the resolution to really high resolution, like 300 dpi, 600 dpi, and so on. So everything is exportable, yes.
Now, Dr. Mistry, how regularly different databases in your talk are updated?
Another great question. So yeah, I have been talking about different databases. I've been talking about Oncoland database from Omicsoft, and I've been talking about Ingenuity database for IPA. So Oncoland database currently is updated on quarterly basis, and in futures, our developers are hoping that they can increase the speed of this update. And IPA database, the information that's curated from literature is actually updated on weekly basis. On the other hand, information that we curate from different third-party databases like BioGrade, clinicaltrials.gov is updated on quarterly basis. So we can, in summary, say that all the major updates are happening on the quarterly basis. And another thing I want to mention over here is we not only update with knowledge and information, but we also update in terms of new features. So a lot of things I showed you, especially in IPA in the later half of the presentation, like bar charts, heat map, and so on, they were not always there. They have been added with all of the updates we've been introducing.
Now, Dr. Mistry, our last question. Is the curated information or data QC'd?
Yes, absolutely. So in terms of land database, it is QC'd at multiple places. So even before we decide to bring data in, we have QC process. Once the reads are mapped to the reference, there's QC process in terms of like how many reads are mapped versus how many reads are not mapped. There's also QC in terms of metadata curation. And in terms of IPS database, there's also a QC process. So first of all, we have PhD level curators curating the information, and this information is spot checked before it's added to the database. So QC parameters are applied at multiple levels before the information is added to this databases.
I would like to once again thank Dr. Mistry for his presentation. I would also like to thank QIAGEN for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through January of 2019. You will receive an email from Labbers letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.